we're going to look at is species and speciation. We're pretty much figuring out how we can determine one organism from another. And we started talking about that with the ibex in the last lecture, where it was, how do we know this is an ibex? How do we know what makes this species different from that species? In order to get that, we have to have a coherent definition of a species. Now, we throw the term around all the time. You always hear people talking about this species or that species, but we really need to look into what defines a species. Now, speciation has a historical significance. Obviously, Charles Darwin talked about the origin of the species. But there's a lot more to it than just Charles Darwin. We talked in the first lecture about how it went back to these ancient Greek roots, how it went back even farther. Humans are big on classifying. Humans are big on identifying and saying this is this and that is that. The earliest works, so our earliest written works with dealing anything with science, we're talking about species, or the specific groups, as individuals with that were similar to each other. I love this. So it's an individual organism represents a group of similar organisms. That was the original definition, translated. The current definition is a group of related organisms that maintains a distinctive set of attributes. It hasn't changed much. It hasn't changed much at all. I tend to trace this back to the idea of the Platonic form. So this is where my whole philosophy background comes into play. Again, how many of you guys have taken philosophy before? Good. You guys have an edge. Did you read Plato's Republic? Because that's the best one. The Republic by Plato had the concept of a form. The form is an idealized, there's an ideal creation that we have in our heads. You all have an idea of what something is in your head when you identify it. For instance, check this out. What is this? A chair. Well done. Thank you. Thank you for being the person in the class that recognized this is a chair. Why? Why is this a chair? What makes this a chair? That's where it gets harder. We look at it and we're like, oh, it's a chair. Why? Because you sit on it. It's entirely defined by its function. So this is also a chair right here because I sit on it, right? There we go. Would everybody else define this as a chair? No. If I was writing on the chair, if I was like sitting here, and you guys have done this, you have your computer in front of you and you're typing on it. Is this a chair now or is it a table? Or is it still a chair because a chair is a chair? A chair is a chair. Table's a table. Except when it's a chair. It's a multifunctional chair table. Is it a chair if it doesn't have a back to it? Maybe. It's a stool then. I know. Well, no, we're defining things because we're humans. Like this table here. If it was, I don't know, shorter, would it still be a table? Yeah? If it was shorter and narrow, or now is it a stool? Could be a bench. What's the difference between a bench and a table? Height and a back, maybe. We all have this idea of what something is in our heads, and that's what Plato is trying to say. There is an idealized form, and we recognize it because in our head we have this form of a chair. We have this idea of a perfect chair, and we say that's chair-like enough to be called a chair. That is table-like enough to be called a table, or a desk, or whatever you want to call it. Do you see the issue, though? We have definitions of things as humans. But those don't always translate perfectly into the physical world. We only have language to support a certain number of objects. Now, just because we label something a species, just because we label that and that are the same thing, doesn't mean they actually are. It's just what our imperfect minds can comprehend at this point. I know that's a little bit deep, and I'm sorry, but I wanted to point out there's a lot going on as far as notation and identification that is more focused on us and our human flaws than it is on the actual natural world. So Aristotle came up with the Scala Natura, where he eventually was the one who identified the words genus and species. So genus means general, species means specific. The genus for him was substance, matter. And then all of matter split into body and spirit, and then body split into living and non-living, and living split into animals and plants, and animal into man and beast. Then that's where we got to the specific. These early philosophers believed that we could recognize a vital life essence in something, a vital essence that was distinct and unchanging, that allowed us to connect what we saw with what we understood it to be. For instance, what is this? What's it an image of? How about that? A lion. How do you recognize it? Well, we recognize it because we say it's got certain lion-like traits. But what the ancient philosophers thought was, this has the essence of a lion. Our essence recognizes that essence, and we say lion. Our essence recognizes the essence of the chair and says, this is a chair. And we're not wrong. So humans got super into, let's just classify everything and name it all. So we could categorize animals by lots of different traits. We could categorize them any way we want, and there was no wrong way. Everyone tried to codify it, but then we just kept doing it all differently. And that gets us to the modern age. Today, we're still classifying things. We're still looking at things and trying to figure out, how do we classify it? How do we put things together? Because in the modern world, there has a serious significance to it. It's financial. If I create a new organism, I as a biologist build something new, I can trademark it if it's a new species. But if it's not new species, I cannot. This bacteria is called agrobacterium. Agrobacteria, let's see, that's A. tumefaciens. There's A. tumefaciens and A. rhizobium. If I use A. tumefaciens to create a new plant, I have to pay the company that owns the rights to this species a lot of money. If, though, I have a slightly different bacteria that is a new species, I don't have to pay rights to anybody. So what's going to behoove me? Use this one that they've already found, or genetically modify it enough that it's now considered a new species. If I modify it enough, I get all the money. See the difference? Speciation is really important for modern pharmaceutical companies. It's really important for modern biotech companies because you can trademark one thing and you cannot trademark another. And if you trademark it, if you own the patent, then everybody who uses it has to pay you money. So what current modern taxonomists are doing is setting species apart by distinct characteristics. It could be morphological traits, reproductive isolation, molecular features, ecological factors, or evolutionary relationships. Each of these is flawed, but each of them has its merits. For instance, morphological traits. What does it look like? That's the big thing. This is probably how we classify things most often. What does it look like? These are angry birds. They're classified together. Which of these is least like the others? Which one? The green one? You guys think this one right here is the most different? Everybody agree? The yellow one? Oh, we have two different opinions, both of which are equally valid. Why would it be the green one? Because it's a long beak, but it does have a round body and long legs. What about the yellow one? Why would not? Because of the triangle shape. You have two good arguments. Who's to say which is better? Okay, what are these here? Not trying to trick you. Fish. What is the easiest way to sort them out? Color. You look at them, you're like, oh, they're different colors. These red ones go together, the yellow ones, the blue ones. That works. Morphological traits are what we typically look at when we're identifying species. We could look at reproductive isolation. If you have two organisms that are isolated reproductively, that is, they cannot produce viable offspring, they would be termed different species. At the top, this is something you don't see often. It's a donkey cross to a zebra, they can't have offspring. So they are two different species. They have offspring. The offspring cannot produce its own offspring. It's infertile. Do you know what they would call that at the top? Donkey and a zebra cross? A debra? A zonkey? I would go with zonkey personally, but debra makes sense. Unfortunately, it's called a zebroid. I know, seriously, who would call that a zebroid? It's a zonkey or a debra. They do. This is a crocoduck. The crocoduck does not actually exist, for those of you who were wondering. It's not like it roams around. But it's the idea that a crocodile and a duck cannot reproduce successfully. It would be really messy if they tried. Probably for the duck. It's important to be able to reproduce offspring. You could look at molecular features. You could look at what DNA do they have, what do their chromosomes look like, what are differences, and how different do you have to be a different species. Between humans and chimps, there is less than 3% difference in our genetic sequence. Less than 3%. You are, and also, I'm pretty sure I'm 3% different than like my cousin. So the big difference occurs on that chromosome right there. All our other chromosomes are really, really similar to each other. So you could look at the chromosomes and say a certain percentage difference means you're a different species. And that's how some people want to do it. Look at ecological factors. Where do they live? Where do they prefer to mate? Here we've got two types of primates. Ones that walk on the land, ones that prefer to be in the trees, the arboreal ones. Typically, the ones who tend to walk upright are going to be classified in one group. The ones who tend to be arboreal are classified into a different group. And that's entirely based not on their molecular makeup, not on who they can mate with, but based entirely on where they live. So this is another factor that can come into play. And you could look at evolutionary relationships. The glyptodont is it's about the size of a VW bug. The armadillo looks a lot like the glyptodont. It's a direct descendant, we think. And you can look at those relationships. So we could say if two organisms descended from the same ancestor, they're closely related. Speciation's hard. There are lots and lots and lots of arguments for and against any of these. And I think we can all admit that none of them are perfect all by themselves. But by combining them together, we can get a better idea of what a species is. At least as good as Plato's, it's a chair, because I looked at it and I know it's a chair. Now we're back to Darwin, because apparently I have to keep bringing them up. Mainly in this picture, I wanted to show them comparing beard sizes. This is Darwin and Alfred Wallace. If you look at them, both of them came up with this idea of natural selection at roughly the same time. Both of them put forward this idea of what is a species? How are these things different? So we have to have a unified species concept that I can go through the rest of this course with you guys on. I need to know how you define a species, because truthfully, there's a bunch of ways to define a species. We need a way to figure out how to distinguish one set of organisms from another. There are problems with all the species concepts I'm about to tell you. What we want, the holy grail of species concepts, is the one that will apply to all organisms at all times. But we need a way to operationalize this. We need it to be able to apply to bacteria just as well as it will apply to this liger. So there are 4 species concepts: the biological species concept, evolutionary lineage concept, ecological concept, and general lineage concept. We're going to go through each. I need you to pay attention, because you have to tell me which one we're going to go with for the rest of the semester. And then I have to rework the lectures. So first off, the biological species concept. Two species are different. They are two different species if they cannot produce fertile offspring together. If we were to take two organisms, cross them together, and they cannot produce fertile offspring, they would be considered distinct species. Makes sense. There are problems, obviously. How would you be able to apply that to bacteria or a protus? Kind of tricky. This up here is a triploid daylily. This daylily cannot produce seeds. It's infertile. It exists. It's totally infertile. It's a cross between one species of daylily and another species of daylily. We put them together, we get this. This, though, can reproduce vegetatively. It can clone itself over and over and over again. So is it a new species or a member of one of the other species? Here we have a wolfen. A wolfen is a cross between a whale and a dolphin. It happens. Whales and dolphins are very different groups. But they can breed and produce a wolfen out in the wild. The wolfen can then mate with either a dolphin or a whale successfully. So which group does it belong to? And that's an issue with biological species. Wolfens? I honestly don't know. That's the question, right? Do you get a wolfen, or is it a whale now, or is it a dolphin? Do we have to look at the molecular data? What do you do? Something like that just came out. I just read an article about whaling in, I want to say it was whaling in Iceland. I know, this is a crazy topic, but whaling is illegal in the North Atlantic unless you're hunting, I want to say, the gray whale. But in Iceland, there's a company that was caught with what looked like a blue whale. And blue whales are endangered species. You cannot hunt them. You can't touch them. You can't do anything to them. So they're doing DNA tests because the company says it's not a blue whale. It's a cross between a blue whale and a gray whale. So therefore, I can hunt it. So they're doing DNA tests to figure out, is it a blue whale, is it a gray whale, or is it a cross? Now they know if it's a blue whale, this company's going under. If it's a gray whale, the company is fine. And if it's a cross between a blue whale and a gray whale, they don't know what they're going to do yet. It's going to go totally to the courts. So these species concepts actually do come into play. You might have the evolutionary lineage concept. In this case, we're going to define a species by its unique evolutionary path. A species is a direct descendant of an immediate ancestral species. Our ancestral species was, I don't even remember what it is. But the idea is, if you go back in time far enough, you will see a distinct shift between one species and the next. So you could cluster groups together that are related through an ancestral species. Now the problem with this one becomes, when do you start defining a new species? Are your parents the same species as you? I'm pretty sure we're all going to say yes, although some of you are actually probably thinking, well, you haven't met my parents. Yeah, they're probably the same species as you. Your grandparents, if you knew them. Same species? Probably. Great-grandparents, you've seen pictures. They look kind of like you. But if you keep going farther and farther and farther back, at some point, there's well, you're different than you. There's a big difference. It may not happen over one generation, but within generations, you can see a difference. So that's an issue with the evolutionary lineage is, at what point do you define a species? Where's that definition? Where's that stopping point? And then the ecological species concept. Populations defined by a group sharing a common ecological niche, or niche. So here is a picture of the thing that I really, really wanted to swat at for you guys. Wasp nest. It's up in a tree. It's big. But that's not where all wasps live. Where else do wasps live? Underground. So you have two groups of wasps. Those wasps can mate with each other successfully. They can produce fertile offspring. They're obviously descended from the same group, but the thing is, one set lives up in trees, one set only lives underground. You guys ever run over a wasp nest underground with like your lawnmower and freaked out when they charged you? No, it's horrible. I'll tell you that. Horrible. Don't do that. You don't know where they are, though, because they live underground. Would you consider the wasps living in the trees a different species than the wasps living underground? They don't mate with each other normally. They can if you force them to. just never run into each other. They just don't get along. Different populations. But are the populations different enough to be considered a species? No. Or yes. Some people think it is. I mean, I'm bringing this up because this is legit in the literature. The last concept I want to bring up is called the general. I agree with you, by the way. Totally agree with you. You're still mating. Well, you could, yeah. Right. They may look different, but we could still mate with them. And that's kind of the way we look at it, right? Same species because we can mate with them successfully. But then we saw this issues with the biological species concept. And it doesn't really gel well with the protists. So last species concept, general lineage. Each species is a population of independently evolving lineages that evolved from a specific series of ancestors and, as a consequence, forms a group of organisms with a particular set of characteristics. So we're right back to, I'll know it when I see it, and also they're related to each other. What this does is it blends all those other species concepts together into one. But in that blending, it also adopts all of their weaknesses. We can't go back and actually see what's related to what. We don't know for sure. We can hypothesize, but we don't know. So here's what I want you guys to do as tables, I guess, sort of like across the row, except you guys in the back, you three, you work together, yeah. Work with the group in front of you. What I'm going to have you do is pick one of these. Rationalize it, and then we're going to come together as a class and we're going to figure out which species concept is the one that we're going to use for the rest of the course in here. As far as what you're going to want to know for your exam, which is the thing that's always on your mind, what's going to be tested. There's probably going to be an essay, and the essay is probably going to be something along the lines of describe the 4 species concepts and justify which one you think is the best. So, take a few minutes, think about it, tell me what you think the best species concept is. We will decide on what it is, we'll decide as a class which is the best, I guess through voting. You guys argue about it for a while, and then I will call today done, and we'll pick it up next time. So we've talked about basic speciation concepts. We've talked about what makes a species. We've disagreed on what makes a species because that is literally how taxonomy works. People get in the room together. They argue over what makes a species. Is that a species? Is this a species? They go to conferences. They write papers. They're very prolific. Now, whether or not we agree on a specific species concept, we do all recognize that there's a lot of differences in the world. We're not all one big unified organism or set of organisms. Although, that being said, there is a sect of taxonomists out there right now who are saying, yes, actually, we are just one big set of organisms interbreeding. So we can't have anything called a species. Here's the deal. There's a lot of variation out there. There's a lot of organisms out there. There's a lot of uniqueness out there. And we get this wide variation in species. What causes this wide variation is usually a very, very sudden event. Some species switches and suddenly has multiple chromosomes, or there's reproductive isolation. A flood, go figure, a flood, right? A flood happens, and in this flood, a single unified population is split in half. Then they start to slowly over time adapt to their new niche. There's genetic drift. You get changes in allelic frequencies until eventually they cannot reproduce. So you have a sudden change in an organism. You could have a sudden change in an organism. And that organism will over time adapt to its specific ecological niche. At first, two separated populations, two populations that are distinct after that sudden event, can still interbreed. They can still mate successfully and produce offspring. So according to our species concept, then, they're the same species. But as mutations accumulate, as they change more and more and more, they're eventually no longer able to successfully reproduce. So first you're going to have some sort of what's called prezygotic mating barrier, so before fertilization, and then after that one comes, then eventually you get total reproductive isolation, mechanisms that stop the molecular mechanisms that stop the egg from being fertilized. What we're looking at when two species diverge is what's called cladogenesis. You take a single ancestral species and it diverges into two species. It's the branching of a tree. Here we have a single ancestral bird, and over time that population diverges into what may look like the ancestral bird and something that looks distinctly different. They have a common ancestor, but they've changed. We're going to create some clades. We can do that with one of three models of speciation. Now, these models are discussed back and forth. In fact, Allopatric and Sympatric are fairly well established. Parapatric speciation is hotly debated right now. I tend to think it makes sense, but I'm just giving you a heads up. A lot of people think that there's really just Allopatric and Sympatric speciation. So we're going to talk about Allopatric, Parapatric, and Sympatric speciation for the inevitable question of what are the differences between Allopatric, Parapatric, and Sympatric speciation. First off, we have Allopatric speciation. In Allopatric speciation, you're going to have some members of a population becoming isolated from the other population and then slowly diverging over time until you have two distinct species. In this slide, you've got a single ancestral white bunny population. Mountains suddenly arise because, you know, they're tricky like that. They suddenly appear, and as those mountains appear, now you have on either side of the mountain a white set of rabbits. Well, it could just happen that on this side, maybe due to natural selection, maybe due to genetic drift, you get changes in the fur color. Until, and on this side you maintain white, until over here you end up with two distinctly different looking populations. Now the question is, have they been separated long enough they won't be able to breed? Because biological species concept, if they can mate together, they're the same species. If they're apart for long enough, then they will no longer be able to breed together. An example of this is the porkfish and Panama. Fun word. The isthmus. How do you say it? Isthmus. 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 It's hard to say. It's awkward, isn't it? It's a weird word. I-S-T-H-M-U-S. Isthmus. You don't pronounce the T? It's just isthmus? Well, that's way easier then. Oh, guys, now we have to find out, do you pronounce the T in isthmus? Isthmus. Can somebody find that out? Crow, are you on that? All right. Important biology things. How do you pronounce this word? What? It's just isthmus? Oh, that is so nice. Thank God. The isthmus. Yeah, that works. The Isthmus of Panama. Appreciate your time. It's this land bridge connecting the two continents. It was underwater at some point. Kind of cool. There's a lot of history to the United States that we don't learn about, really, because it happened before there were a lot of governments involved. It was just indigenous people and water. There was a sea. All of Mexico and Panama was underwater. It was a vast, huge, shallow sea. And over time, you have mountain upwelling, and you get Mexico and Panama built. Now, Panama was one of the last places that came up out of the ocean. Before that, there were sea currents that could go between the Atlantic and the Pacific Ocean, and it changed the entire atmosphere. You have wind going over the top and ocean currents going between them. Life was very different. And then came Panama. Once Panama arose, you have this species called the porkfish. Now, the porkfish is swimming around. in the Atlantic Ocean and the Pacific Ocean that were connected, suddenly they are no longer connected. You have two distinct populations. Here's the Pacific version, here's the Atlantic version. They look kind of alike, but over time, as that land was there, they could no longer breed, so they diverged. And then humans came along, and we said, we want to get through that with boats. So what do? We dug a really long canal through it. You may have heard of said canal, the Panama Canal. So we dig through there. Now the pork fish actually can start getting back and forth to each other. But turns out they don't mate anymore. If they don't mate with each other, are they distinct species? According to our biological species concept. If they cannot mate with each other, they are distinct species according to that concept. So we have allopatric speciation, a sudden event. It may not have been that sudden, but the rising of Panama separated this population, causing them to no longer interact for a long period of time. Several rounds of allopatric speciation can lead to what's termed adaptive radiation. Adaptive radiation is where one single ancestral species evolves into lots of different descendants that differ greatly. Here we have some sort of balloon animal horse thing. And over time, its descendants have adapted to their niches, leading to distinct variation. Whether they're long-necked, long-horned, long-tailed, short-necked, whatever. You have one species that then radiates to fill specific niches. This happens a lot after an extinction event, a mass extinction. When you suddenly have a whole lot of organisms wiped out, anything that's left will radiate outward, will radiate and fill those ecological niches, or niches. We'll talk more about the importance of adaptive radiation when we talk about history of the Earth. Darwin's finches describe adaptive radiation. Single ancestral species that fill different niches on different islands. Where two populations meet, two distinct populations meet, and they can reproduce, they form what are known as hybrid zones. Here, two populations can interbreed freely. We've got a blue population, a purple population, a black, cream, and a teal population here. Where purple meets blue, there's going to be a hybrid zone. There you'll see some organisms there that are mixes of the two species. Let's see. I think it's in Virginia. It's usually in southern Virginia. There's an issue with both wolves and coyotes. Wolves and coyotes typically do not share the same range because they're top predators. But where those two populations meet, you end up finding what are called coy wolves, a cross between a wolf and a coyote. Because though we may look at them and be like they're different species, they look at each other and like, I can either mate with it or I can eat it. And that's life. So they mate. They produce an offspring, and that offspring has traits of both parents. That's termed a hybrid. You will find more hybrids in the hybrid zones than you will, say, out in the farther ranges. If you were to take a bunch of hybrid zones and put them edge to edge like this, where you've got blue will mate with purple, but blue will not mate with black because blue and black don't ever meet. They're too diverged. So you got blue mates with purple, purple can mate with black, black can mate with cream, cream can mate with blue, but blue cannot mate with teal. See how that works? There's no zone connecting them. There's no hybrid zone. So that's allopatric speciation, where some event happens, some barrier occurs, something stops, some physical barrier stops a population from mating. Sympatric speciation occurs in the same range. So with allopatric, you split the population. With sympatric, it happens in the same population. So you have one interbreeding population that suddenly has this weird thing inside of it that isn't able to reproduce with anybody else. So we got our white bunny. We get some allelic variation introduced. So you have a white group and a brown group, say. They can at first freely interbreed. They produce hybrids in a hybrid zone. But over time, it may occur that that white rabbit and brown rabbit can no longer mate. That's where you get dead hybrid. The hybrids can't survive. As long as hybrids are viable and they survive, according to the biological species concept, these are the same species. But once those hybrids can no longer survive, then you have two distinct species. Now, this is really tough on the biological species concept. Again, back to the coy wolves. Wolves are one species, coyotes are another species, right? Oh, wait, except they can produce fertile offspring. So there we go. Sympatric speciation occurs within the same range. This is very predominant in plants. It's not surprising to see a field of one flower with a totally new species originating inside of that field. One way this happens is through polyploidy. Do you guys remember polyploidy from last semester? What does it mean to be polyploid? Oh, never mind, it's right on the board, right? You have two or more sets of chromosomes. Not two or more, has more than two sets of chromosomes. Diploid organisms are termed euploid. That means they have the right number of chromosomes. You are diploid. Your gametes are haploid. If you have 3 complete sets, or 4, or 6, or 8, then you would be termed polyploid. Now remember, what are chromosomes made of? Not trying to trick you. DNA. DNA wrapped around a bunch of peptides. DNA is going to produce the plans, RNA, to make what? Proteins. So if you have 2 copies, you may be making 2X proteins. If you have 3 copies, you may be producing 3X proteins, which is 1.5 times more proteins than you need. That could be an issue because your body is highly regulated. You need to have the right number of proteins at any given point. If the organism survives the polyploidy, though, suddenly they're going to look really different. With polyploidy, one mechanism of polyploidy is called alloploid. Here an organism has at least one complete set of chromosomes from 2 or more different species. Okay, that's weird. That's like you mating with a gorilla and producing something new. Animals don't do this well. Don't try. I'm not like endorsing that. But plants do this a lot, where you can get one parent, big A, big A. It's got its whole, a full genome complement. It's diploid. It receives pollen from this other parent, BB. It's got a full complement, a full genome. It's a different genome. And it produces an offspring that contains the entire genetic complement for both plants. Now, the only way this can happen is when what's called a non-disjunction occurs. Did you guys learn about non-disjunctions last semester? I don't know how far the other teachers went. Let me explain what a non-disjunction is. It's going to be hard because I don't have anything to write on the board today. All right. Puppet show. So, you guys know remember meiosis and mitosis? Okay, good. Whew, we're set there. So, in meiosis, you've got chromosome. Good on that? All right. At some point, these are homologous pairs. So what'll happen is they're going to replicate. So you have an exact copy of each. So far, so good? They form tetrads. Awesome. Okay, we're in the same place. These tetrads are supposed to split apart and then split apart again, leading to a single chromosome in each gamete. Does that make sense? We're good? What ends up happening in a non-disjunction is same thing happens, chromosomes, they replicate, you form tetrads. The tetrads, though, only split one time. So instead of getting one chromosome into each gamete, you get a full complement. You got 2 chromosomes, twice as many as you should have in a gamete. But exactly how many you need to have a complete copy of the genome. If that happens in 2 plants, and their pollen fuses to the egg, then you're going to end up, the sperm fuses to the egg, sorry, the pollen splits. Then what you'll end up with is an offspring that has a full complement from one parent and a full complement from another. It will be really super different. It is no longer going to be able to make gametes that will match parent A or gametes that match parent B. It's all on its own. That's okay for plants, though. Some plants. Why? With us, that'd be terrible. If you end up being, and it's not going to ever happen, but if hypothetically, some animal ended up having was alloploid, 2 complete sets of genomes from 2 distinct species, it would not be able to interbreed with anything else because there's nothing else like it. But why can plants do this? Thinking back on Mendel and stuff. Yeah. It's okay. No. You can totally look. The platypus thing was really esoteric. You want to take a shot? No. Eva? No. Are you sure? Yes. It clones it. Okay. So yeah, plants can clone themselves. Vegetative reproduction. You can expand outward from the roots. But even more than that, when you're fertilizing a flower, does it absolutely need another flower all the time? No, it can do it to itself. We can't do that. No matter how much you may try, you're never going to be able to produce an offspring with yourself. Again, not trying to endorse you trying that, but there you go. So you got alloploid. You can produce completely different offspring that have these unique genetic characteristics. Alloploidy leads to absolutely abrupt reproductive isolation. There will be no reproduction with any of the parent species. It is all on its own. And reproductive isolation leads to speciation. So both of these, alloploidy and polyploidy, are major mechanisms for speciation in plants. You can go from a diploid organism here to a tetraploid organism here. They look very different. They cannot reproduce together. Okay, another way you get sympatry. In a geographic area, you're going to have a lot of variation. And certain organisms can adapt to certain niches. So here, we might have mangoes and bananas growing in the same area. It is a single geographic region. Some fruit flies may like to mate on mangoes. Some fruit flies may like to mate on bananas. By the way, if you've ever wondered why these organisms are so attracted to rotting fruit, it's because they love alcohol, which is what the fruit's producing, and they love to mate. And so they're pretty much, that's where they go to mate, is on your food. Now you know. Great way to collect these guys is to literally go on a beach with a bucket of rotting bananas, sit out there for the afternoon, let them fly to you. You have a margarita or something. They collect in the bucket. You put a top on the bucket. You walk to the lab. That's that. If you ever wanted to spend a grant really quickly. Hypothetically speaking, that is. These may be right next to each other. These flies may shoot past each other. But they will never mate together because they choose to mate on their specific fruit of choice. Same geographic region, but they're in 2 different areas. That leads to speciation. They're not mating with each other. Over time, they cannot mate with each other. And we're back, I think, to that ecological species concept. Are they 2 distinct species? If they're 2 distinct species, then they can't mate with each other. One, that's biological. But also, they just happen to never really run into each other. And then mechanism number 3 is sexual selection. Yeah, okay, so usually it's the female that screws this one up. The female chooses males in general. Across the animal kingdom, that's the way it works. Homospecific mate choice. What does it mean to be homospecific? What does homo mean? Same. Specific is specific. So it means you're pretty much choosing the things that look like you. The more you choose something that looks like you, the more you're going to get two groups diverging. Here we have two species of fish, P. pundamilia and P. nairii. They look different, and it turns out the reason they look so different is because the female pundamilia chooses to mate with the lighter-skinned males, and P. nairii chooses to mate with the darker. They're looking for that coloration when they're choosing their mates. They are absolutely distinct in the wild. If you were to go out there, the females will not mate. You can't force them to mate with each other. But once you change the light, once the female can no longer discern the difference between one male of a species and a different male of a different species, she'll just mate with anybody, and it turns out they can produce fertile offspring. Now, in the wild, they tend not to mate. They live in the same exact region. They live in the same habitat. They swim next to each other. They're eating the same food. But due to female mate choice, they are speciating. According to the biological species concept, they are the same species. According to the ecological species concept, they're different species. But this only happens because of who the female wants to mate with. And then the last model of speciation is Parapatric speciation. Here we've got a very, very, very large range that's contiguous, as a contiguous population. That means that on this end there are bunnies, and on that end there are bunnies, and there are bunnies all in between. There's no barrier separating them, just distance. Now this is where it kind of gets weird. It's all about logic. This is a giant brown rabbit. We're good on that, right? We'll call this one A. A can mate with B. It's a slightly smaller reddish-brown rabbit. They're kind of similar. They can mate with each other. They produce fertile offspring. Are they the same species? No? Well, according to our species concept, biological, right? They can mate with each other. They're the same species. Moving down the line, we've got this sort of smaller rabbit and this larger brown rabbit, slightly different colors, but they can mate with each other. They produce fertile offspring. Life's good. They got this cool hybrid zone in the middle. So they're the same species. Moving farther down, these guys can mate with each other and produce fertile offspring. These guys can mate with each other and produce fertile offspring. These guys can mate with each other and produce fertile offspring. So A and B are the same species. B and the C are the same species. C and D are the same species. D and E and F are all the same species. Yet, F cannot mate successfully with A. Therefore, A and F are different species. So it's sort of saying A equals B, B equals C, but A does not equal C. Super fun, right? It's both logic and sort of math and biology all wrapped up, which is why people are falling asleep. That's Parapatric. You get what's called a cline. So on one end, you have an extreme phenotype. On the other hand, you have a different extreme phenotype. And in between, you get a slow progression from one to the other. So that is one species, this is one species, and these in the middle are something else. The Corvus crow. The East Coast has pretty small crows. The West Coast, so our crows, we look at them and we may think they're big, but our crows are only about, that big, about a foot. On the West Coast, they've got gigantic crows. You look at them, you're like, that's not a crow. That's some sort of buzzard or something. They are messed up looking. So for the longest time, we thought they were distinct species. Turns out, genetically, they are the same species, just with different sizes, different phenotypes. When you get a whole bunch of species in Parapatric like that have distinct hybrid zones in between them, they might wrap around a geographic barrier. This is called ring speciation. You've got brown, orange, green, blue, pink, and down here purple. Blue can mate with pink, pink can mate with brown, brown can mate with orange, orange can mate with green, green can mate with blue, but blue cannot mate with brown. They're the same species according to the biological species concept, except at the same time, they're not. It's a logical inconsistency. This actually happens out in California. You end up seeing these ring species around large deserts, surrounding the edges of them. Now, we've been talking about speciation as if it happens very, very quickly, and it doesn't. Or it does. One of the two. There are two distinct models for speciation. The gradualist model and punctuated equilibrium. Again, for those keeping track, we've got Allopatric, Sympatric, Parapatric. What are the differences between them? That's a big concept. New concept we're going into is differences between gradualism and punctuated equilibrium. In the gradualist model, you go from a single ancestral species, and over time, that species may slowly shift. Its morphology will change very slowly. So at first, the differences are slight, and they get more and more extreme. Snail shells, for instance. Snails are generally aquatic. These aquatic snail shells here represent a shift from a tidal region that has a lot of wave action to a more freshwater or slow-moving water form. So in case you guys, just sidebar, snail evolution, super interesting. As these shells get thinner, squatter and squatter and squatter, what you're seeing is a difference in water action. Have you been to the ocean before? Played in the water? Waves? Yeah, okay. Waves are heavy and hard, and they carry all sorts of sand with them. If you've ever, say, ridden a wave or gotten knocked over by a wave, and you got up, your pants were filled with sand. Maybe not. I mean, I may be the only one that's happened to. I come out of swimming in the ocean, and literally I've got, because guys get pockets. I had pockets that were filled with sand and debris. You know, you're just pulling it all out. Not to mention, any other place on your body that sand could possibly get into, it gets into. Happens to snails, too. Snails are even smaller than you, so if a wave comes in and hits them with all of that sand, it sort of knocks them around. These shorter, stubbier shells withstand waves much, much better than long shells. Long shells, though, allow the snail an adaptive advantage in freshwater. One, because they're pointier and can hurt things that try to step on them or eat them. But the big thing is, these guys, the species C is getting an advantage in rough water. Species B is getting an advantage in freshwater. So they're slowly diverging due to natural selection. That's gradualism over a long time. Punctuated equilibrium, on the other hand, shows rapid changes. There are long periods where everything stays about the same. And then suddenly, in the next period, everything has this new form. What could cause such a sudden rapid shift in the fossil record? Now they're just snail shells still. What would cause snail shells to go from being really, really round to really long in a very short amount of time? A bottleneck. So what you're saying is there's something that comes through and just wipes out most of the snails. That could very, very much be it. Or something that has a strong selection, a super strong selective event. Not even a bottleneck, but say, I don't know, a new sandbar formed, or a bay was cut off, and so suddenly now there's no more wave action going on. Without that wave action, that stubbiness doesn't have any advantage. So very, very quickly you end up with much longer shells. So punctuated equilibrium is pretty much you stay the same for a long time and then super quick change and now you're something new. It does not happen in one generation. It could take many generations, but it's not over thousands upon thousands upon thousands or millions of years. There are three factors that will impact the speed of evolution. Allopatric and high natural selection. If you have an allopatric event where mountains suddenly spring up, or a river forms, or population is split by some physical barrier, that will speed up the change within that population, because suddenly you have two distinct allelic frequencies now that are diverging with either drift or something else. High rates of natural selection. Let's say in this room, I don't know, aliens come through and they wipe out anybody not wearing yellow. So pretty much everybody's dead except for Courtney. I'm sorry, Courtney, sorry, except for Rachel. Rachel, is that your yellow drink too? Did you totally coordinate? Yeah. Awesome. Except for Rachel. Rachel survives. She goes on to reproduce because apparently she can do that asexually. And now everybody's got yellow. It's a sudden shift. It makes evolution happen really, really, really fast. A high mutation rate combined with environmental shifts. If an organism is constantly mutating, a bacteria, and the environment is also shifting, we're adding new antibiotics. There's a high rate of evolution. Over time, the allelic frequencies in that population will change like crazy and lead to a reproductive advantage. And generation time. Bacteria reproduce every, 4 hours, 5 hours. So you can split and split and split. Yes?
Two questions. One, what is that demon spawn on the top picture there?
That up there. It does not, it actually is worldwide. That is a frilled shark. A frilled shark is an early shark form that we thought was extinct. Turns out it's not. It lives close to the bottom of the ocean. It will come up to the surface when it's sick. They caught one recently, kept it alive in an aquarium in Japan, I think, and then it died. But the frilled shark, we have a lot of images of. It is a basal shark form. It has not changed its shape or its form in millions upon millions upon millions of years. The reason it has not changed its form is because the bottom of the ocean hasn't changed much. So slight, no environmental shift leads to a low rate of evolution. So that's what the demon spawn is at the top. Frilled shark. Here, the bacteria, fast, very, very fast generation time. Elephants, very slow generation time. They carry their offspring for more than a year. Then their offspring don't reach sexual maturity even longer than that. Ooh, turtles, tortoises don't reach sexual maturity until they're like 60 or 70. I mean, it's a long time. That's from one generation to the next is 60 or 70 years, which means they don't have the opportunity to have their alleles affected as much. There's not as much mutation or migration added to that changing frequencies in that population compared to bacteria that are everywhere reproducing all the time. So between these three, you can affect how fast evolution happens. It can happen quickly. It can happen slowly. Now, there's some evidence for evolution in what's called evolutionary developmental biology. Evo-Devo bio. It's the development of different species is examined in order to understand ancestral relationships. The idea behind this is that organisms, as they develop, are going to display traits that are indicative of its ancestors or of who it may be related to. As these things develop over time, here you can see a fish, salamander, tortoise, chick, hog, calf, rabbit, and human. Early stage of development, we all look kind of similar to each other. As we progress through development, you can see changes happening. Between the fish and the salamander are probably more closely related than the rest. because these guys look about very similar. These guys look similar. I don't know where I'd put you. And these guys look similar. Further along during development, you could see even more differences. We have the fish and the salamander, probably very closely related, as are the tortoise and the chick. So evolutionary developmental biology is really concerned with, can we draw relationships between the developmental stages of different organisms in order to see how they're related to each other. Now, this does not take into account what are called back mutations. It does not take into account unique variation in development. It doesn't take into account that some organisms actually revert to an earlier primitive state. What evolutionary developmental biology is more concerned with now is the gene expression, when certain genes are turned on and when they're turned off. Some genes will affect development and alter phenotype dramatically. Usually these are called Hox genes, H-O-X. Write that down. I don't have it on the board. Hox genes. Hox genes are developmental genes. Hox genes determine whether you have a left side and a right side, a head. They're your body plan genes. And by changing them just a little, you can radically alter your body plan. We're not going to do that. It's not something scientists are looking into. They're not like, let's mess with this and see what happens. It was likely changes in the Hox genes that caused a proliferation, an explosion of life during the Cambrian era. So we originally had these Hox genes develop. We got a body plan, left side, right side. And then through mutation, you end up seeing lots and lots of changes. Now you don't just have 4 limbs like we do now. You had like 8, 15, 32 limbs, whatever. Instead of having 2 sides, the left side and the right side, you'd have 5 sides or 3 sides. It's kind of weird when you think about it, but it happened. As you look more recently, just slight differences in Hox genes will cause variation in the shape of a foot of a duck and a chicken. During development, they look exactly the same. It's only when you get the genes that turn on to clip webbing that they're different. Take a look at your fingers. You'll notice that there's a slight web between them. That wasn't always, your fingers were originally fused all the way to the top until this gene kicked on that clips them down. Some people have that webbing all the way up still because their genes never turned on. Usually that's corrected with surgery, but it's all about turning on one gene at a particular time during development. If it's turned on at the wrong time, then the organism ends up being completely different. Now, some organisms, humans included in this, are going to display completely different body arrangements or relative body sizes between their juvenile form and their adult form. A juvenile human has a gigantic head. You notice that? Look at any like kindergartner and you're like, whoa, they're like bobbleheads. They're huge compared to us. We've got much smaller heads. They've got very, very large heads in proportion to their bodies. Now, this is just kind of, what if a baby was the same size as an adult? You know, that's what this graph is showing. And you can see the different relative head sizes, different relative arm lengths. Just this morning, my wife was concerned if her skirt was too short to go to work. And I said, well, we can always do that test that they do in school where, what is it, you have to like have your fingertips down to your side. And if your fingertips are at the edge of your dress, then it's fine, or something like that. And she did it, and her fingertips were way high. And I was like, see, it works out. Well, the reason for that isn't because suddenly it's a different length. The deal is, as adults, we have shorter arms in comparison than kids do. It's the way it is. They're growing at weird rates. You guys remember puberty? Some of you guys were just there recently. It's awkward. You got your limbs growing at these different disproportionate speeds. At some point, you probably had super long legs, or like your arms were really long. Your feet were growing insanely like you went from having like normal size or normal proportion feet to having clown feet. I mean, it's the way it is. That's called heterochrony. Different proportions for different body sizes at different points during development. Now, some organisms are kind of cool. They go through what's called pedomorphosis. Pedomorphosis is where you're going to retain a juvenile characteristic if it gives you an adaptive advantage. So the adult still retains some juvenile character. Did you know at some point during your development you had these things called pharyngeal slits in your neck? They allowed water to come in and out so you could process it, sort of like fish do. It's true. Now, if you at some point underwent a pedomorphosis event, that would have been retained into adulthood, and then you could breathe water. How cool. This salamander, it happened to, an early salamander. It's called an axolotl, I think, now. Thanks, appreciate it. That's an axolotl, isn't it? So it's got this Pokemon look to it. Closest thing you can get to a Pokemon. The axolotl is an aquatic salamander. Now, salamanders, usually in their adult form, come on land. They're amphibians. You have an aquatic juvenile form and an adult land-based form, just like with frogs. This one decided, and it's not like it decided, there was a mutation. And the genes that were required to convert the gills into lungs never turned on. So it retained gills into its adult form and continued to reproduce. It still had gonads. It still was able to reproduce. So now we have this whole new species that looks distinctly different from the normal salamander. Now, don't worry. That's not like don't worry. The axolotl is actually extinct in the wild. It lived near Mexico City. There was a lot of
one weird lake near Mexico City.
And they paved over it. Yeah. They're like, it's over. So the axolotl only exists in aquariums now. I mean, there are a lot out there. It's just it doesn't exist in the wild. So there we go. That's pedomorphosis. Over time, one organ or one trait can be adapted to fill new niches. Now, people always look at the eye and they say, well, the eye is too complicated. It could never have formed on its own. It could never have given you a fully fleshed out eye. It just makes no sense. There's no intermediate forms. But the deal is there are. You can go from one form, photoreceptors, just light and dark. That's kind of important. Now you can see, so as life evolved, the evolution of the eye was really, really important to the evolutionary arms race. Because if you can see light and dark, suddenly you know where, where light is, but more importantly, if something was to pass over top of you, you could see shadows. You would know that there's something coming. Over time, that absolutely gives you a reproductive advantage, right? You see something coming, you can move. That was big. Over time, if it became concave, has a little indentation to it. Now you can figure out direction of the light. Is it getting dark over there? Which way is the shadow moving, and how fast? As you become even more concave, you be concave, and you get this layer growing over the top. Now what you're getting is a pinhole camera effect. The light is being reflected is coming down and angled very, very precisely. So now you're not just getting what direction it's coming from, how fast it's moving. Now you're also getting shape distinctions. Adding a refractive lens, once it's completely covered, you can still have light coming through, but you've got a fluid-filled section in here that helps to give you maybe color vision. You could distinguish more lines better. The eye is very complex. It can absolutely, it looks marvelous. The whole human body is a marvel. Just the idea that all these parts work together and we wake up and we're alive each morning. I mean, there's so many things that can go wrong, it's not funny. Don't go into advanced biology or you will realize how much can possibly go wrong and how freaking amazing it is that we're not just waking up not a pile of ashes. It's phenomenal. But the eye, as an evolution, can happen. It's happened multiple times. Our eyes are evolutionarily distinct from mollusk eyes, the eyes of a squid. Structurally, though, they're exactly the same. In fact, in a lot of labs, people will dissect octopus and squid eyes instead of mammal eyes because it's interchangeable and they're cheaper. They've got the same exact structures, but they came about in totally different ways. Do you guys remember what that's called? Two distinct evolutionary lineages producing the same structure? I think I mentioned it once and only once, but I'm not sure. They are analogous, and analogous structures lead to convergent evolution. They result from convergent evolution. Two structures that look very, very similar because they're adapted to the same niche. Okay, so that is basics on the speed of evolution and what could cause evolution to happen. Now, for our biological species concept, you have two distinct ways of stopping these organisms from mating. Prezygotic sexual isolation and postzygotic sexual isolation. What do you think prezygotic means? Before what? The zygote. That's right, yeah. Before the zygotes. This happens before successful mating. Things that stop successful mating. This is the Brazilian sea slug. The Brazilian sea slug is very interesting because it goes to mate. It wants to mate with other sea slugs, obviously. In order to do this, the Brazilian sea slugs get together in a group. And the reason they get together in a group is because they've put out this literal sea slug aphrodisiac. It causes them to go into a mating fury. They just sort of get together and mate with anything that looks remotely like a Brazilian sea slug. That has the proper chemical scent to it. Unfortunately, if a Brazilian sea slug was deficient in that aphrodisiac, deficient in that chemical, the other sea slugs wouldn't play with it. It would like miss all the reindeer games, you know? It's totally excluded. Just gets to sit there and watch. That's no fun for it. It wants to mate like everything else. The field cricket. Crickets chirp. Perhaps you've had one in your house and it's kept you awake. They chirp not through their mouth, they chirp through their legs, but they play a very particular very particular song. If they play the wrong song, nobody will mate with them. You guys all have the preferred music, right? You prefer some sort of music to another music. And there's some music that you just completely judge people on. I mean, I'm correct in this, I assume. You're just like, oh, you listen to that. Those are people you're probably not going to mate with. It's prezygotic isolation. Before they even have the chance, it's over. Prezygotic isolation happens prior to insemination. Now, this is a bird doing a mating dance. Birds display like crazy, trying to get the attention of other birds. If they dance wrong, the other birds will not mate with them, and it's over. They're isolated. There are 5 prezygotic mating mechanisms. Habitat isolation, temporal isolation, behavioral isolation, mechanical isolation, and gametic isolation. There are examples throughout the animal kingdom, and of course, examples in humans. For instance, habitat isolation. There's a geographic barrier that makes interbreeding impossible. Something is stopping you from mating. Some mountain is in the way. Some vast distance. You live in a different place. Anybody had ever tried a long-distance relationship before? Did it work? It did? 4 of them? For 4 years. Oh, no, that's a ring. That's a ring. Sorry. Well done. The rest of you guys, not so much, right? No, it didn't work. It's really, really hard, and you can attest to this. I'm not asking you to. It's really hard to mate with someone when they're in a different city, in a different state, different country. If they're somewhere else, you cannot physically mate with them. That barrier is stopping you from mating. Habitat isolation examples. If you are, back to the mangoes and the bananas, these guys prefer the mango, these guys prefer the banana, never the two shall meet. They have been stopped from mating due to that isolation. Here we've took two flies, a single population, we split it. This one was given maltose to eat, that one's given starch to eat. Over time, they, again, remember, flies breed on their food. So when you see flies landing on your food, they're ready to mate. They may be mating, they may be laying eggs. It's all about food, and sex go hand in hand with flies. So here, they've got that maltose food, they're laying their eggs in it, they're growing up in it, their larvae crawl around in it, they hatch, they mate in that food. Several generations later, you take them out, you put them in the maltose group and the starch group together, and what you end up finding is that the starch ones only mate with the starch ones, and the maltose ones only mate with the maltose ones. You've isolated them, you bring them back, and now they're two distinct species. You split a population of beetles, you've isolated them from breeding, they can end up speciating. They are not able to reproduce with each other. Temporal, yes?
So that's
Just like the pork fish. Allopatric is usually run by habitat isolation. Temporal isolation, time. Due to timing of the mating cycle, it'd be hard for two organisms to interbreed. You cannot go back in time right now and mate with somebody who's already dead. Makes sense, right? Or even weirder, it would be difficult for you to mate with somebody who is significantly older, because they may not be able to produce offspring anymore. The mating cycle was off. As far as other organisms go, frogs, there are two kinds of frogs, spring frogs and fall frogs. No, spring frogs and summer frogs. The spring frogs mate in spring, the summer frogs mate in summer, as the name implies. If you're a spring frog, you're not going to ever mate with the summer frog because your times don't overlap. You've got to time it properly. Cicadas are probably the best example of temporal isolation. Do you guys remember cicadas? They come out every now and then and it sounds really loud. So there are 1 year, 3 year, 7 year, 13, and 21 year cicadas. That means that some cicadas come out every single year. They dig down, the eggs hatch, they dig underground, they eat dirt for a year, they come out, and they crawl up a tree, emerge from their shells, and start flying around mating. They live for about 2 or 3 days, and their whole job is mate. They don't even have mouth parts. They can't eat. Their whole life is mating at that point. Then they die. What? It's okay. No, it's okay. What? It's okay. So those 1 year ones can mate with each other. The 3 year ones, though, can mate with the 1 years and the 3 years, but they will only ever mate with those. The 7 year ones won't ever see the 3 year ones until year 21, so they will only mate with the 7 year ones and the 1 year ones. They're isolated from the other populations. When you hit that magic 21, there was a few years back, I think it was probably about 12 years back, there was the magic number where they all emerged at once, and it was the loudest thing you've ever heard. It was like science fiction lasers going off all the time, all night, for a few weeks. Crickets. If the cricket's not... I got a funny cricket story for you. If the crickets cannot mate with the other crickets that are sexually mature at the same time they are, it's over. I mean, that's just two distinct populations. fun cricket story. So back, just so you guys know, the way I previously taught 102 was I would work with each individual student and have them design their own labs. So that instead of doing paper labs, it was students would work to pick a project, pick a problem, and solve that problem in the semester. Then they'd write the problem up and present it. One of the students had just transferred from Liberty. Now, Liberty, as you guys probably know, is a very conservative school, and he sort of lost his faith. So he came and he decided to prove that God didn't exist. And I was like, I want to see where this goes. So his idea, and you guys can easily tear this experiment apart, I just let it go, was to take crickets. He took crickets and he put them in a box. And he said, all right, I've got crickets in box 1, my control, and in box 1 there will be, they'll feed the crickets. Makes sense, right? These crickets will do fine. Box 2, he will not give those crickets food. Instead, he will ask people to pray for them. And he was going to show that apparently God doesn't like crickets. So he had a controlled experiment, and I was like, all right, let's see where this goes. And he's watching them. Oh, no, wait, I'm sorry. He did actually several different other boxes. Control, he'd have people from like his former church pray here. He had like other people from other denominations pray for this box. It was cute. And I was like, all right, there we go. So what ended up happening was, over time, he was watching, dead set of his experience, and all of them survived. And he said, he was shaking to the core. It was great. And he's thinking all these things, and he wrote it all up. And his conclusion was, God exists. And then I wrote and said, I was like, well, actually, I tore his experiment apart because you were testing something that couldn't actually be tested. But it turned out I just fed the crickets. I just wanted, because that's exactly what I would do. I'd totally mess with you guys that way. Anyways, so the kid ended up going back to Liberty, I think. Newfound hope and faith. And never will he know. So that's the cricket story. Otherwise, do you guys know what happens if you leave a bunch of crickets in a box with no food? They eat each other. Yeah, they die because they eat each other. There's one fat cricket at the end. Behavioral isolation. Different populations may have different ways of signaling their prospective mates. As you can imagine, it is hyper important to properly signal a mate that you would like to mate with them. You've been in these situations before where you didn't know if somebody was flirting with you. It required your friends to tell you. They're like, they're flirting with you. You're like, no. It's not the way that works. They're doing it wrong. They need to come over and say, I am interested in a relationship with you. That's the way that, yes, okay. Behaviors. You expect one thing, they expect something totally different. You need to make sure the signals are working well. I think that's how I ended up, one of the reasons I ended up with my wife was because I was pretty sure she was interested, but I wasn't totally sure. And then she said, I'm interested in beginning in a relationship with you. And I was like, that's a good signal. I agree. We should do that. I think we were probably in a relationship at that point. I'm pretty sure. I wasn't totally sure, though. And it's not like I was doing anything else. So it's like, okay, this is good. We're communicating on the same wavelength. Birds. Birds sing, and they're like, tweet, tweet, tweety, tweet, 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 which to another bird means, I am ready to mate with you. We hope. The deal is, though, birdsong changes. Birds either sing to mate or they sing to declare their territory. So if the birds get their song wrong, I feel bad for the bird who doesn't understand signals. For us, it's just awkward. For birds, it's like, hi, I'm here to mate. And the other bird's like, no, I was warning you away. I will kill you now. And you've seen that. You've seen those two little birds attacking like the giant birds. That's behavioral isolation. if they are not able to sing the right song, not only are they never going to attract a mate, they may attract the wrong kind. The insect mating dance. I did a lot of research on fruit flies. I swore I would never deal with fruit flies again. And I'm probably going to end up dealing with fruit flies again. But for now, I've been safe. Fruit flies have a very complex mating stance. And if the male fruit fly screws it up, the female will utterly reject him. I mean, it's all female choice. So what ends up happening, so let me describe the mating box that I had for fruit flies. This was my life for a while. I would sit down in a lab, and there's a big clear box and a little sock that I could put my hand in with you guys have seen pipettes at this point, right? This pipette had a tube, and that tube went into my mouth. You can see where this is going to get bad. So I put the flies in, and I would catch mating couples. So after I saw them go through these motions, I would have to suck up the couple, put them into some food so they could lay eggs, and I'd count the eggs and things like that. So I put it in, I go, and usually what would happen is the fly would go into the pipette, and I'd, into the food. But then I sucked too hard, and it went right down my throat. So I was like, ooh, that sucks. That's when I put a little cotton swab at the end. But yeah, if the male doesn't dance exactly right, he's got to land next to the female. So female flies. Female comes in, the male comes in. He gets sort of where she can see him because the female really doesn't want him like in her blind spot. That's just creepy. So he's like, hey, check me out over here. And he's like, I'm coming over here. Now check me out over here. He does that 3 times in this particular species. I'm over here. I'm over here. I'm over here. Do your eyes like me? And then he starts to sing. And the way the fruit fly sings is they flap their wings at a certain vibration. And he has to hit exactly the right vibration. If it doesn't hit the right vibration, the female's like, no. No. You don't sound right. You sound stupid. So the female will then either fly away, or if she's still interested, she will start walking away and expects the male to follow her. So at this point, the male is in her blind spot. She can't see him because her eyes don't do that. He has to lick her in exactly the right way. If he doesn't, it's over. That leads to copulation. And then when that's finished, finally, that's where the female's like, and okay, get off. I have things to do and food to eat. Oh, speaking of which, don't forget this all happens on their food. If the male does any one of those steps wrong, if he doesn't land in the right place, sing his song right, if he just gives up, she's like, oh, she's leaving, I'm going. Oh, and you guys have done that. You've done that before, right? Like somebody's done the whole they're walking away, and you're like, oh, okay, they're not interested. You've never done that. You've never followed. You're like, wait, maybe. No, it's over. If he licks her wrong, if he doesn't copulate, if he's incapable of copulation, it's over. Now, after that, obviously, he can be rejected any which way she wants. Humans, eye contact. Eye contact means different things in different relationships. For me right now, just so you know, I'm making eye contact with you so I can figure out if you're actually paying attention. See? But in some cultures, eye contact is threatening. You don't make eye contact with someone because that is asserting dominance. Yet in other situations, you might be flirting. And this is where it all gets weird, doesn't it? It's like you're out, you're looking at somebody in the eyes, and you're like, do I have something on my face? Is there something? Oh, you make eye contact, and then they scratch their nose, and you're like, does that mean that I have something on my nose? Or does that mean they just had an itch on their nose? I better subtly check. And then you do that, and then they scratch their nose again, so then they're thinking, is there something on my nose? There's a lot that goes on with behaviors. You can have mechanical isolation. Question? Or just stretching? Okay. There can be morphological features on the organism that stops 2 species from interbreeding. Literally, there's something there stopping copulation from occurring. These two dogs, there's an issue. There's a mechanical isolation issue. The little male would have a very difficult time mating the much larger female. A very hard time. I mean, maybe if she bent down. But that's mechanical isolation. The parts don't line up. Here we have these two copepods. This copepod has a cover over its genitals, and so does that one. The problem is the covers don't match up. So they can't mate. You know, snakes, when they mate, male snakes have multiple penises. Not a surprise. Why? Because it's freaking hard to mate if you're a snake. You have no limbs to hold things in place. You just got to get things lined up right. Mechanical isolation. And then you could have gametic isolation. This is sort of the farthest along. In this case, the gametes are incapable of successfully fertilizing. So you have mated. Congratulations. You've got all the right signals. You've got all the parts lined up. Everything's great. You got the sperm and the egg, and they meet, and nothing happens. That's gametic isolation. The gametes are isolated. All of these are considered prezygotic isolating mechanisms because it happens before the zygote forms. Typically, prezygotic mating isolation will occur much faster than postzygotic. We're going to talk about postzygotic in a second. It's far easier to change behaviors than it is to change molecular characteristics. Yes?
Going back to the snakes.
Yes, back to this, okay.
Yeah, so you said they have like multiple penises.
Yep.
Would it be possible if they made with
Yep.
one?
It's possible, yep, snake orgies. They call them mating balls. Not kidding. Sorry, guys. Welcome to biology. What ends up happening is you'll see a lot of snakes in one particular area, usually like under a rock or under a log or something, connected in different ways. It happens. Yes. Yep. Like people too. So, I mean, it's totally possible for a female to have two eggs down at one time and two different males at two different points. You can end up with twins, fraternal twins from two different fathers. Was it Jerry Springer? Yeah. Here's a gametic isolation example. These are, what are you guys? You're sea anemones, I think. Urchins? Anemones. So here's the deal. Back when I was teaching in Greensboro, we had a whole tank of these guys because we absolutely used them to collect their sperm and their eggs. The way you can collect sperm or eggs from a sea urchin or a sea anemone is you take saline and you inject them with saline, and that causes them to release all their sex cells at once. Now the deal is, in the ocean, these guys don't like jump on top of each other and start mating. Instead, what they do is they will just release their sex cells and they'll go on the currents. And then as two sex cells from compatible species meet, that's where you have the zygotic fusion. So it's sort of just like, let's see if we can get this to happen. Usually what will happen is you'll have a whole group of them expelling their sex cells all at once so that there's a higher chance for success, but it might happen differently. We would do this in order to, like I said, collect their sex cells for the meiosis lab. So you could see actual fertilization occur instead of just, I don't know, imagining it or whatever we do here. Kind of neat. Oh, that did lead into a fun discussion, though, because I had everybody, they had eggs and they had sperm, right? And they took the eggs and the sperm and they put them onto the same slide. And they're looking at them under a microscope. And the students were very, they were as interested in as you could be in biology. So half of them were caring, half of them were just like, I want to get through this. And we got to the end of the lab, and I said, okay, clean everything up. And so they poured them down the drain. And then I said, okay, now let's talk about in vitro fertilization. And they're like, oh, I just watched something alive down the drain. It's like, yep, there you go. Gametic isolation examples. Here is a sperm, a bunch of sperm, not able to get into an egg. Then we get post-zygotic sexual isolation. This occurs after the zygote has been formed. So in this case, we've got a horse and a donkey. They mate together. They produce a mule. The mule, though, is inviable. It's incapable. I'm sorry. It's not fertile. It's sterile. It's incapable of producing offspring. A goat and a sheep cannot produce fertile offspring. With post-zygotic sexual isolation, Haldane had said, he's a population scientist who came up with the idea that when in the F1 offspring of two different animal races and one sex is absent, rare, or sterile, that sex is usually heterogametic. So that means if sterility occurs, it will occur first in males for humans. The heterogametic sex, the one with the X and the Y, or the Z and the W, depending on what mechanism we have. So if something's going to happen, it's going to happen probably to the male. Why? Because the female has another X to make up for the difference. That's also known as the large X effect. Males become sterile before females. Hybrid inviability will occur, so that's sterility. So you got a horse and a donkey make a mule, and it's sterile. Inviability is where the organism cannot survive to be born. You take an egg and a sperm, the zygote doesn't develop beyond early, a few cell divisions. It just dies on its own. So you have hybrid sterility and hybrid inviability. And these are ways of post-zygotic isolation. These reinforce speciation. These tend to be very strong at hybrid zones, where two populations meet. So inviability is they die, sterility is they survive, but they cannot produce offspring on their own. So pre-zygotic isolation comes before post-zygotic isolation. And I've got to figure out a way on the exam to word that so that people don't just say, well, pre comes first because it says pre. Pre-zygotic mating isolating mechanisms, like behavior changes, happen much faster than post-zygotic ones. So if you have these two populations that were formerly separated by a pre-zygotic mating isolating mechanism, you bring them together, and they're no longer able to successfully interbreed. Now you have two species according to the biological species concept. You've changed some signal. You've changed something about them that caused them to speciate, that caused them to become something new. So it's thought that pre-zygotic mating isolating mechanisms are pre-zygotic pre-zygotic isolation has to come first. This is a necessary step to causing two species to diverge. Everywhere we've seen this in experiments, it's always been pre-zygotic isolating mechanisms happen before post-zygotic. This sort of helps with the biological species concept because the reduction of fitness of hybrids can, and eventually lack of viability, leads to two distinct groups that cannot produce offspring. So here's the one thing I can throw you as a bone to support the biological species concept. Any questions? So you got a bunch of review questions from this one. So describe the four species concepts, evaluate them all for validity. Sounds like a great essay question. Describe the 5 types of pre-zygotic sexual isolation. Another pretty good one. Understand differences between pre- and post-zygotic isolation, and describe differences between punctuated equilibrium and gradualism.